So we're not going to do the anaplerotic reactions, which are great, and we're not going to do the glycosylate cycle, which is also great. Sacrifices must be made. We'll talk about the citric acid cycle, variably and interchangeably called the tricarboxylic acid cycle. Citric has 3 carboxylates, or the Krebs cycle. Okay. This is where, again, this is the oxidative step. This is where complete oxidation occurs of the carbons of glucose. When we switch over to metabolism, I said it's about taking those electron-rich carbons in CH and CC bonds and getting them off of those carbons to make CO bonds in terms of CO2. We should remember that for our purpose, for this purpose, for aerobic glycolysis, the oxidation that occurs in glycolysis counts, right? So fermentation preserves NAD under anaerobic conditions. Under aerobic conditions, fermentation does not occur, or at least doesn't have to occur. So those NADs that are made aren't regenerated, the NADHs that are made aren't regenerated, they're kept, so they're used. So that degree of oxidation counts. Already a little bit oxidized when we're going into the citric acid cycle. And so this is going to be about following the electrons. If it's redox reactions and electrons are being transferred, where do they go? Okay. In terms of carbons, the carbons are input into the citric acid cycle in the form of acetyl-CoA. We'll talk about acetyl-CoA a little bit. We said it's got a nice, great, big, wonderful structure, but for our purposes, that's the piece of it that matters. Coenzyme A has a thiol, acetyl-CoA has a thioester. So it can be, so it's acetyl for that 2 carbon, but it can be acylated with anything that has a carbonyl. So we can see, formyl or propanil or whatever. And of course, pyruvate is 3 carbons. With a methyl group. So we can kind of see which parts of the pyruvate molecule can get appended to the coenzyme A to make acetyl-CoA. And then that would imply then that a carbon dioxide, CO2, which again, ultimately every glucose, if it's aerobically metabolized, is going to produce 6 CO2s, right? 6 carbons need to be fully oxidized. So in the formation of acetyl-CoA, because there's 2 pyruvates per glucose, 2 of those 6 CO2s can be accounted for. And it's accounted for with what we can generously call an enzyme, but what is really a whole cluster of repeated enzymes. The whole complex is called pyruvate dehydrogenase. But that a little bit undersells what this thing is, because it's actually quite large. It's got dozens of individual polypeptides in it that really catalyzes formation of pyruvate into acetyl-CoA, but in order to do this, it needs to partition that, what I just said, into 5 different steps to make the acetyl-CoA. In terms of eukaryotes, there's also a partitioning that needs to occur in terms of subcellular location. Because the glycolysis and gluconeogenesis occur in the cytoplasm, but the citric acid cycle and oxidative phosphorylation occur in the mitochondria. So the pyruvate dehydrogenase complex participates in getting the mitochondrial substrates in the mitochondria and keeping the gluconeogenesis and glycolysis substrates in the cytoplasm. Now we of course are going to fill in the structures, but if you look at the overall schematic of the citric acid cycle, one, it's a cycle, so it doesn't have a discrete endpoint the way the glycolysis does. End up back where you began. But it still requires an input of carbons. So if we think of it as every 2 carbons from an acetyl-CoA that go in, corresponding 2 CO2s come out. So now we've got the CO2 stoichiometry set, right? So glucose goes to 2 pyruvates. Pyruvate dehydrogenase goes to acetyl-CoA. There goes 2 of the CO2s. There's the next 2 that gets you to 4. There's the next 2 that gets you to 6, right? Because pyruvate goes through this twice, all 6 carbons net are accounted for. Those 2 carbons are added to a 4-carbon molecule, oxaloacetate. 2 plus 4 is 6. And it gets decarboxylated once to 5, once again to 4, right? So the intermediates go up to 6 carbons, and then go down to 4 carbons in time for that to cycle back through. There's substrate level phosphorylation that occurs. So an ATP equivalent is made for every acetyl-CoA, so 2 per pyruvate. And there's also reducing equivalents that are made. 3 NADHs and 1 FADH2 per acetyl-CoA. Acetyl-CoA has 2 carbons, right? So if there's 2 carbons, that's 4 pairs of electrons that can be transferred. Total, to totally oxidize it. So the net is going to be 2 times as many reduced cofactors. How many carbons times 2? So 2 carbons go into acetyl-CoA, 4 net cofactors come out. And well, let's scale this up. So that's for 1 pyruvate, and then there's also there's NADH made for another pyruvate as well. So if there's 6 carbons in glucose, then the net output combining glycolysis, pyruvate dehydrogenase, and the citric acid cycle for the 6 carbons in glucose to be converted to 6 CO2s, there's going to be 12, right? 2 times 6, 2 pairs of electrons, 12 total reducing cofactors being made. And we'll talk about why one of them has to be an FADH2. Try to remember why FADH2 is meaningful, right? That's the thing that has a higher standard redox potential. It can oxidize more highly reduced bonds, right? NADHs are great for converting carbonyls and alcohols. They can't handle alkanes to alkenes. There's not enough energy. It doesn't have enough draw of electrons. FADH2 can handle that single bond to double bond, carbon-carbon single bond to double bond chemistry. But NADHs and FADH2s aren't the end point. They're just another place for the electrons to go, and the electrons have to go ultimately to make ATP. That's what oxidative phosphorylation of the electron transport chain is. So we look at this again in terms of separation of the 2 processes, right? There's a process where the CO2 is converted, the glucose is converted to the 6 CO2s. That's the oxidation. And then oxidation creates a pool of reduced electron cofactors. And then that pool of reduced cofactors can be used in a separate process, electron transport chain, oxidative phosphorylation, to make ATP. So they can be decoupled from each other. And that's essentially what's being demonstrated here in the matrix. So fatty acids, glucose, amino acid can get pumped into the mitochondria, form acetyl-CoA to make these things. The electrons are going into the electron transport chain, which establishes kinase bond gradient, which drives the formation of fresh ATP, of novel ATP from ADP. Don't worry, we're going to go through this in so much detail, you'll know exactly what's going on. But this is the OTP, right?